Hi, my name is Rutendo Sigauke. I'm a student at the University of Colorado Anschutz Medical Campus, and today I'll be presenting a poster titled Regulatory Network Inference Using Nascent RNA Sequencing Data. So gene regulation is controlled by transcription factors, or TFs. They do this by binding it at enhancer and promoter regions, recruiting RNA polymerase to promoters, and driving the transcription of a target gene. Previous experiments have shown that when TFs bind to these enhancer regions, short bidirectional transcripts are produced at these enhancers. These transcripts are called enhancer RNAs or eRNAs. And these eRNAs can be used as a measure of enhancer activity as well. And importantly, enhancer activity has been shown to be specific to developmental stages, cell, and tissue types. And essentially, this ensures proper cell fate and function. In this toy example, I'm showing enhancer A being only active in neurons, enhancer B only active in oligodendrocytes, and enhancer C only in microglia. And so there are a lot of methods that have been developed to link enhancers to their targets. For example, histone modifications have been used to identify active enhancers and active promoters. Other methods include looking at accessible DNA and essentially looking at accessible distal enhancers and accessible gene targets as well. However, both these methods are low resolution and low throughput, which brings us to nascent RNA sequencing. And so nascent RNA sequencing actually gives a unique perspective since it shows transcription at the distal enhancer, giving rise to a bidirectional enhancer RNA, as well as transcription at the target gene. And so this allows for better resolution, high throughput, and cost-efficient readout of both enhancers and gene transcription. Different studies have looked at interacting enhancers and genes in 3D space, and essentially looking at the transcription of these enhancers and genes on the left, which is looking at cage data, and on the right, looking at gross-seq data. What they found is that when enhancers and genes are interacting in 3D space, their transcriptional levels are actually highly correlated. And so this suggested that we could link enhancers to target genes based on the transcriptional levels of the enhancers and genes. So the goals of this project are to build transcriptional regulatory networks using nascent RNA sequencing data. And by looking at publicly available and published available data sets that span various tissue types and experimental conditions, we could identify highly correlated enhancer gene pairs. And we hypothesized that transcriptional regulatory networks derived from nascent RNA sequencing data will result into more accurate and high-resolution transcriptional regulatory networks compared to steady-state assays such as RNA-seq. And so to that end, the Dowell and Allen labs manually curated 2,880 nascent RNA sequencing experiments from the Sequencing Read Archive. And these samples were treated with various treatments, over 300 treatments, and they spanned over 150 different cell types and tissues, and they came from 20 organisms and over 6 different nascent RNA sequencing protocols. And so the goal now is that now that we have different transcriptional readouts from various cell types and tissue types, we want to identify highly correlated enhancers and genes. And essentially taking a module of highly correlated enhancers and genes, we can build networks that essentially tell us how enhancers are regulating gene transcription. And so if you step back and look at the enhancer RNAs that are supported by nascent RNA sequencing data, what we found is that over 20% and even up to 60% of current databases such as GTEx, ENCODE, Phantom5, and Enhancer Atlas actually overlap enhancer RNAs in our database. And when we step even further and look at enhancer gene pairs that are supported by SNPs from the GTEx project, we found that these pairs were highly correlated compared to enhancer gene pairs, random pairs from the database that we have. Essentially, this just supports our findings or previous findings that interacting enhancers and genes are highly correlated in transcription. And so on the right, I'm just showing one example from highly correlated GTEX pairs. And so this is a bidirectional transcript that is on chromosome 3 that overlaps a SNP associated with the TGF-BR2 gene. And so building an atlas of transcriptional regulatory elements will allow identification of enhancer gene pairs and essentially help us get a better understanding of cell type specific enhancer activity. Furthermore, nascent RNA sequencing data provides a robust signal at both enhancers and genes, making it a good tool to narrow in and zero in on enhancer gene activity. So the Nascent database, or DB Nascent, contains over 2,800 samples, and we've collected a lot of metadata associated with these samples. Furthermore, we've processed the samples using a standardized pipeline, and we've collected different metrics for each sample. And the goal is to have transcriptional regulatory networks for human and mouse data in the database as well, giving us a better understanding of how eRNAs and genes interact in different conditions. So with that, thank you for coming to this poster presentation. I'd like to thank the Dowell and Allen Labs for their input and feedback for this project.